Transformation is a process in which a cell is altered so that it can acquire and express genetic material obtained from the surrounding medium or environment. The most common use for transformation is the introduction of recombinant plasmid DNA into bacteria. Bacterial strains can either be naturally transformed or can be made competent by artificial means. Transfection, on the other hand, is the introduction of foreign DNA into the nucleus of eukaryotic cells by non-viral methods. The most common gene transfer methods are chemical, lipid, and physical methods. There are many different methods to transform cells. However, not every method can be applied to both prokaryotic and eukaryotic cells. To transfect eukaryotic cells, electroporation and sonoporation are commonly used. Electroporation is a physical transfection method that consists in the application of electric pulses to create temporal pores in the cell membrane. It can be used in bacteria and eukaryotic cells, and it's the most efficient technique. Sonoporation can also be used as a transfection mechanism, and it is an ultrasound-mediated cell membrane permeabilization, or induction of membrane pores using ultrasound. The pulses of ultrasounds are delivered by a machine, and there can be of selected intensity and duration, depending on the sample. There are several things that must be considered in order to select a bacterial transformation method. First of all, it's important to consider the method through which the cells have become competent. For example, when making chemically competent cells, the use of calcium chloride is required, and high salt concentrations in the medium will damage the electroporation equipment and the cells, so heat shock transformation will be the best option. Also, the transformation efficiency required is something that should be considered, because electroporation can provide higher efficiency than the chemical transformation. Another thing that should be considered is the cell membrane and the wall composition, because of some cell components can be resistant to certain transformation methods. The ideal method for cells that present resistance to other transformation and transfection methods is electroporation, because it facilitates the pore formation in the wall by the electric pulse. Another important factor to consider before choosing a transformation method is the size of the plasmid that will be introduced into the bacteria. Electroporation is a good method for high molecular weight vector transformation. Vectors with a molecular weight of 50 kilobases could be transformed by this method. Finally, it's also important to take into consideration the capacity of the cells to resist the damage caused by the transformation method in order to ensure that the cells will be alive after the transformation method. Apart from the most common transformation methods like heat shock and electroporation, there are other methods that can be used, like bioballistics, that is a mechanic method that works like a gene cannon, in which DNA is wrapped in gold or tungsten particles, then it's disposed in a machine that uses compressed air to introduce the pellet in the cells. Plate transformation, which is a method that consists in preparing a culture of receiver bacteria and incubating it at 37 degrees and 200 rpm overnight. Afterwards, they must be translated to a new medium and incubated at 37 degrees and 200 rpm for 2 or 3 hours. 1 milliliter of the culture must be centrifugated and resuspended in 200 microliters of LB medium, mixed with 10 nanograms of plasmid, and finally speculated in cold plated with calcium. Colony to lung transformation, which is a method used when a plasmid is in a bacteria and it's necessary to introduce it to another one. This method avoids the destruction of the plasmid that is necessary to transform competent bacteria. This process needs to take the donor bacteria and dissolve it in water and ethanol, add calcium chloride and mix with the receiving bacteria, and then transform. There are differences between the type of competent cells when using topo vectors. For cloning and transforming in this vector into chemically competent cells, it is necessary the addition of sodium chloride and magnesium chloride to the ligation reaction to get a final concentration of 200 micromolar of sodium chloride and magnesium chloride. That increases the number of colonies over time. For cloning and transforming into electrocompetent cells, cell concentration must be reduced to 50 micromolar of sodium chloride and 2.5 micromolar of magnesium chloride in order to prevent arsenic. For this, it is necessary to make a dilution to achieve the proper salt concentration. E. coli and some other enteric bacteria possess in their genotype LAC-C, a gene that encodes beta-galactosidase, which is a tetramer that hydrolyzes lactose into glucose and galactose. Some bacteria have been modified to have a mutation which deletes them from the 11 to 41 beta-galactosidase amino acids, so the produced enzyme is a non-functional one because it is unable to form a tetramer. Of the alpha peptide to make beta-galactosidase functional is called alpha complementation, and it can be used to screen E. coli colonies for the presence of inserts. X-gal can be added to the medium for blue-white screening. X-gal is an analog of lactose, so that it can be hydrolyzed by beta-galactosidase and a blue product will be formed. For this type of screening, the MCS, which is where the insert will be placed, must be in the LAC-C region of the vector. As a consequence, when a piece of DNA is ligated into the MSC, it disrupts the alpha peptide, making the beta-galactosidase non-functional. When a bacterial strain transformed with a ligation reaction is spread onto agar plates with X-gal, two different colonies can be obtained. A blue colony that indicates that the alpha peptide in the plasmid is intact, which has no insert, whereas a white colony indicates that the alpha peptide is disrupted and the insert is present.
First, a mixture was prepared adding 3 microliters of PCR product, 1 microliter of salt solution, 1 microliter of the topo vector, that is PCR 2.1 topo, and finally the volume was adjusted to 6 microliters with water. Then the sample is incubated at environmental temperature during 10 minutes. 3 microliters of the ligation is transformed with 50 microliters with the chemical or electrocompetent cells. After transformation, 300 microliters of soak medium were added, and then the sample was incubated at 37 Celsius degrees with shaking. Then all the sample obtained is cultured in a LB plate with X-gal. After 12 hours of incubation, the LB plate was observed, finding only one blue colony formed in the surface of the plate. After analyzing the LB plate with the culture sample, there was found only one blue colony. That means that the plasmid was relegated and the beta-galactosidase gene was expressed.
The cloning with topoisomerase technique is a useful tool because it helps to reduce time by making unnecessary the use of oligonucleotides and restriction enzymes, and it provides a visual alternative for selecting transformed cells beside the antibiotic resistance. It is important to pay special attention to special details like the reagent's concentration in the reaction and the recovery time the cell will need after transformations. Since the vector used will be treated with topoisomerase, it will have a more relaxed structure which will need bigger pores in the cells and more time for it to recover its shape after transformation. It is also important to use high-efficient competent cells that will facilitate the entrance of the relaxed plasmid and will ensure a successful transformation.